that you might not be aware of. And hopefully that brings you some enlightenment and some hope into the future, okay? All right, so moving on. I'm just going to talk a little bit about some cancer statistics. I always do that in any type of talk, and I think we'll kind of breeze through that quite quickly. But then I really want to spend some time with cancer immunotherapies and some personalized therapies that are here and available in Victoria, and some new therapies that are very soon to arrive here in Victoria. And then I want to talk about some of the work that we're doing here at the University of Victoria that I think is appropriate to talk about because of climate change and because of our warming planet. And surprisingly, that has something to do with cancer as well. So I'll give you some information on that, and that's the last thing. And as an introduction to the warming of our planet, I'll talk to you a little bit about cancer metabolism, because I think that's something that isn't talked about very much, especially in the lay media, and I want to give you some information on that as well. Okay, so a little bit about statistics. I think that you might be aware of these things. I think the biggest thing that I'm drawing to your attention is at the bottom of this slide is the website called cancerstats.ca. It's a live website that is dynamically changing all the time when statistics change in the country. You might be aware of some of these websites that developed, especially during the pandemic, to give you information about the spread of the pandemic and about how incidence and prevalence was changing in different communities. So cancerstats.ca, it's a dynamic website, and any type of cancer you're interested in Canada and in any province. And besides these rather grim statistics that are up here, where 1 in 2 people will have some sort of cancer in their lifetime, so something that affects us all, on the next slide is something that I think is probably a little bit uplifting, in that in British Columbia, the darker the blue shows the more prevalence in cancer in Canada. So Ontario and Quebec are much more adversely affected than we are in British Columbia. So we're lucky enough to live in relatively less cancer here in British Columbia than in Ontario and Quebec. And that might be because of the higher population. Certainly. That's actually age and population standardized. But this next slide is really quite interesting, and I want to spend just a little bit of time talking to you about that, and talking to you about a watershed moment that happened about 6 years ago. So I spend a lot of time, I'm an iron biologist. I do a lot of iron biology, and all of the work that I do is in anemia and in iron excess and iron deficiency. And you might be surprised why that's important in cancer. It's because iron deficiency increases your risk for cancer, and iron excess also increases your risk for cancer. So that's a little bit more about me. But the reason that I wanted to spend some time talking about this slide is that you can see that in terms of all of the things that affect Canadians, cancer is one of the things that affects approximately 30% of the population. So that's a large percentage of the population. So we're a developed country. And I said there was a watershed moment that happened about 5 years ago. And that watershed moment was when, in the world, the World Health Organization realized, or the WHO realized, that infection was no longer the unfortunate mortal killer of people on the planet. So as developing human beings, infection was always the most untoward things that we had to worry about. And even in underdeveloped countries in the world today, infection is the most insidious thing that they have to worry about. Now, as you can see, heart disease is also a big problem in Canada as well, and we're a developed country. In underdeveloped countries in the world, infection is still the biggest problem that they have to worry about. And one of the biggest infections that the world has to worry about is malaria. So, and I spend a lot of time working on patients who have a slight resistance to malaria, and those are thalassemia and sickle cell patients. Unfortunately, they're at more risk for cancer. But I want to go back to that watershed moment just for a second and talk to you about that. This is a big change to how the WHO handled their approach to disease in the world when they realized that cancer and heart disease were the two biggest things that were starting to affect human beings on this planet. But it was also a message to us that things were changing. So 2,000 years ago, on this planet, infection was the biggest risk. Now something's changing in the world, and that's our modern developed society. Yes, we live more close together, but there's a built society that we live in. And as we live in this built society, we increase our risk for these types of cancer. We increase our risk for cancers and heart disease, okay? So that's just an idea that I wanted to drop on you, and I'm going to come back to that at the end of the talk, okay? All right. So just wanted to talk a little bit about some of the things that cancer isn't. Firstly, cancer isn't just one disease, of course. It's a collection of different types of disease. And that's why, really, there isn't one silver bullet or one type of therapy that really can control cancer. There are lifestyle changes that kind of give us a hint of what these environmental things are. There are lifestyle changes that we can make in our diet and our nutrition, and in terms of how much exercise we do and things like that. But I'm sure you've heard people talk a lot about that in the past. So I wanted to talk, as I said, about things that you probably might not be aware of in terms of cancer. And really, cancer cells, of course, are not one type of cell either. Cancer is not one type of disease. There are many types of different types of cancers, and they are all relatively different from each other. Although there are some therapies that I'm going to talk about that can be very helpful to many of these types of cancers, but they are different types of cells even. So what we really know now about cancer is that they, even though they might develop from a cell that's gone through a mutation, for example, we now know, of course, that there must be multiple mutations. It's not one single event that causes cancer. Although there are exceptions to that rule, and those exceptions to that rule might be when you are exposed, for example, to a high dose of radiation. For example, if you were working in Chernobyl in 1986, unfortunately, you probably got leukemia if you didn't die in the days after. The Fukushima unfortunate disaster that happened to Japan, after that unfortunate earthquake and the tsunami, the damage at that the radiation leakage there increased the risk for leukemia. So these are different types of things that are unusual events. Most people get cancer as they're aging, so cancer is also a disease of aging, although there are leukemias, of course, that affect very young children. And I want to talk to you about one particular type of leukemia that we've done a lot of work in at the end of this talk. One other important thing about cancer cells is that, as I've said, they acquire different types of mutations, many types of mutations. They're not they tend to despecialize. They are not doing the job that that cell usually does in the tissue anymore. So they're not, so for example, if it was liver cancer, for example, or if it was lung cancer, those cells would no longer perform the job that they were intended to do. And they become younger in nature. They become highly prolific and they start to divide rapidly. And I want to talk to you about some of the ways that we can control that as the talk goes on. And the other thing I'd like to say is that the cells in the tissue where cancer is growing in is very much like an unresolved wound. And wound healing, actually, is a new type of research that is very informative to how we understand cancer. Cancer is like an unresolved wound, where some environmental effect is unresolved, and a lot of the wound healing that goes on as these cells become highly prolific is very similar to a wound healing situation. And on the next slide, I've just got some ideas that you've probably heard of in the lay literature. Some of these mutations that happen to the cancer cell, in other words, the genes are damaged. Some of the genes that are damaged are in specific genes that will cause them to become highly proliferative. We call those oncogenes. I'm not going to talk too much about this anymore. And some of the other genes are called tumor suppressor genes. So those are genes or parts of our DNA that actually suppress this highly prolific type of growth, but they become damaged and not able to do that. So they've lost their functionality, and so the cancer isn't suppressed. One of the biggest problems that we see in cancer research and in cancer in general is the unfortunate problem of immune suppression. And a lot of what I want to talk to you about today will be ways to get around immunosuppression. Your immune systems are fantastic systems, but they become suppressed in cancer. And that's why immunotherapy is so strong right now and is giving a lot of hope, I think, in society today, to leukemia patients, to melanoma patients, and to other types of leukemias as well, that I'm going to talk a little bit more about at the end of the talk. This slide seems quite busy, but I want to talk to you a little bit about what's going on in the slide. Up at the top of this slide is a typical environment, a tumor environment, where a tumor will start to grow. As I said, it's like a wound healing process. The one thing I want to give to you as information is that not only does cancer really start in one cell by one specific event, but it's also caused by not one cell itself. In other words, there are other cells in this environment that contribute to the cancer, and I want to give you more information about that. And this is important in understanding cancer and also about understanding therapy to cancer. So not only does cancer originate and develop in one cell, it depends on the other cell types in the tissue. So in the lung, in the liver, and in the pancreas, and in your brain, it's not only made up, so the liver is not only made up of liver cells, for example. In the lung, it's not only made up of lung cells. There are other supportive cells in there. And those supportive cells, like fibroblasts, and like immune cells, and like fat cells or adipocytes, they're also very important to normal cell function. But when cancer starts to arise, it's just not that one cell that causes cancer. The other cells in that tissue contrive together to cause the cancer. So it's not just developing in one cell. The fibroblasts, the adipocytes, the immune cells, the endothelial cells as part of the blood vessels, they're all contributing to the cancer, to the development of the disease. So new therapies that are coming out now target those other cells as well. So I think there's a lot of hope in that arena of targeting those other cells in combination. The other thing that is very interesting, and I would say relatively new, that you're probably not really aware of, is that when a cancer metastasizes, or when a cancer moves from its primary location to another site, and that's the really sinister part about cancer, isn't it? When it metastasizes. It was over 100 years ago that we realized that cancer spreads like this, and that's the insidious problem in cancer. But we didn't really know why. For example, breast cancer might gravitate into the lungs or to the bone. Prostate cancer is famous to becoming a part of the pelvic bones, for example. Why does cancer spread to a specific target? So we now know some of the fundamental answers to that question. And on this slide, you can see that the primary tumor, when it moves to its metastatic site, the primary tumor is releasing other things besides the cells that travel to that metastatic site. It's releasing factors that we can measure in the blood now. It's releasing small little vesicles that we call cancer exosomes. You don't have to remember that. You can ask me lots of questions later on in the morning, right, about cancer exosomes, or about these tumor-derived soluble factors, or these TDSFs that are up here, those little colored triangles up at the top. These tumor-derived soluble factors, we can measure them in the blood now, and they tell us a lot of things. So that's an exciting area that's happening right now. And there are lots of other factors that are actually released from the bone as well. There are hematopoietic or bone-derived factors that are released from the bone that will actually lead the cancer cells to that area. So this is a really ripe area of research that is really exciting and might be a really profitable and hopefully, when I say profit, I mean a really ripe area for new therapies and really successful therapies to stop this metastasis from happening. So we can go on to the next one. And so for the rest of the talk, I really want to talk about what's going on in the immune system. The immune system is at first very effective at getting rid of cancer. Every day, if you don't have cancer, your immune system is working to remove unusual cells, and it does a great job. The problem is, in 10 or 20 years, in this so-called equilibrium phase of this, what cancer researchers call cancer immunoediting, this equilibrium might be very long. It could be 50, it could be 30 years, where there is this dormant phase, where the cancer just sits there. There are new mutations growing inside here, and then it escapes. The problem that happens here to allow this escape, in a large part, is immunosuppression. And I want to talk to you a little bit about some of the ways we can overcome this now. We can pass over the slide, and I can talk to you about that later. In personalized therapy, in the past, we gave one single type of therapy for many different types of cancer. Radiation therapies, cisplatin, different types of chemotherapies would be used for all sorts of different types of cancers. But now with rapid sequencing techniques, we can become, and different biomarkers that are now available, some of the things that I was just talking to you about, these soluble tumor factors, some of these circulating tumor cells that are, we can now pres prescribe precision medicine that is more specific, and instead of causing unwanted adverse events, you can have benefit to specific types of cancer in specific populations of people. So how do we do that? So that's what I want to talk to you about, more precision, and the first area is in immunotherapy. So this has been around for about these two types of therapy, a little bit more information that we can talk about later. We can make our immune cells active for longer. That's the first thing that we can do. And this is very effective. This made a huge splash about 10 years ago, and now is very effective. The other thing that we can do on the next slide is that we can reprogram T cells, or our specific immune fighter cells, with specific ways of engineering those cells. We can take T cells from a patient, re-engineering them, and we can actually do that here in Victoria right now. We have a brand new lab at the Cancer Control Agency and the Cancer Research Center here in Victoria, and they can make what is called CAR T cells. These CAR T cells, or these chimeric T cells, can be re-engineered to fight specific cancers. And that's available here in Victoria now. The other thing that we can do is that we can, on the next slide, is bispecific antibody therapy without even taking anything out of your body. We can amplify your immune system and bring those T cells to fight specific cancer cells. So that's the next thing. I was telling you about those circulating things that are released from the cancer. So there are circulating cancer cells that we can analyze and improve therapy. And there's actually, on the next slide, there's circulating DNA, or circulating genes in your blood that we can take a blood sample and we can analyze that. And now in Canada, on the next slide, without even going to your physician, there's a new test that's available. It's a $2,000 cancer blood test, and you can order that test now. You don't even have to go to your physician, and that test will come back showing you your risk for cancer. And I can talk to you more about that. The last thing that I want to talk to you about are three old guys that were in Europe in the 1930s. Otto Warburg, Hans Krebs, and Christian Bohr. I don't have too much time, so maybe we can talk about this a little bit later. But they were all, between the 1930s and the 1940s, scientists in Europe. Otto Warburg discovered that cancer cells take up more glucose than any other type of cell in the body. And this was because of hypoxic areas in the tumor. And we've spent a lot of the last 5 years at the University of Victoria working on this, and we're very interested in that. The other thing that I think is hugely important is that Christian Bohr, at the same time, realized that hemoglobin in the blood is dependent, and the affinity of hemoglobin to oxygen is dependent on both the temperature and the amount of carbon dioxide in the blood. And this has been used by exercise physiologists for over 100 years in that understanding. When you exercise, you produce more carbon dioxide because your body is more metabolically active. Some of those tissues actually become hypoxic. You produce more carbon dioxide, and it allows hemoglobin to release more oxygen. So that's standard exercise physiology. As you exercise, this helps you release more oxygen at those muscle cells. What we didn't realize is that, and I'll give you a little nutshell picture of this, is that the environmental temperature is directly related to the amount of metabolism. So as the temperature goes higher, you increase your amount of metabolism. You increase the release of carbon dioxide, and that affects the amount of oxygen being released by the tissues. If you don't exercise, and if you don't live at higher temperatures, if you have lower levels of carbon dioxide, you release less oxygen at the tissues and they become hypoxic. So this is a bit counterintuitive, but we thought that was really important. So on the next slide, I just wanted to give you some information of what we found. As you increase temperature, the prevalence of leukemia in Europe, this is one of the hottest in climate change. Europe is increasing in temperature faster than any other area of the world, about approximately half a degree faster than here we are in North America. So as you know, the general environmental temperature is increasing in the world. And we found that the prevalence, or also the incidence of acute lymphocytic leukemia, is directly related to the temperature in the environment of countries in Europe. And what we did over here on the right-hand side is that we also realized that as the temperature is going up between 2009 and 2020, that the incidence of leukemia is actually going up as well as climate change is going up. So I just have a couple of seconds left, and I just wanted to go to the next slide. I could probably draw to your attention again that website, cancerstats.ca. We talked about immunotherapies, CAR T-cell therapy, about all these circulating markers that are on our bloodstream right now. The new test that you can either order right now, you don't have to go to your physician to get that. And the prevalence of ALL, I'd be welcome and happy to talk to you about and how that is correlated to increased environmental temperature in the world today. So thank you very much for all your time. I think I've gone a little bit too long, but we can talk more about this later on this morning. Great. Thanks so much.